Hi everyone, thanks for watching. I'm Piao. I am a postdoc researcher at the AI.CRED Institute at KAIST, working with Professor Sung-Soo An and Professor Woo-Young Kim. Today I'm gonna talking about our recent project called AdaPert, which is about predicting how cells respond to perturbations. So during my PhD at Seoul National University, I was doing graph-based machine learning methods in clinical and biomedical domains. I worked on many real-world problems like patient state prediction using clinical notes, molecular property prediction, and drug response prediction. For drug response prediction, the setup here is that we have a gene expression profile, which is basically a big matrix of cells by genes. And we also have the drug structure as an input. And the model tries to predict IC50 value that measures how sensitive is the cell to the drug. But there is the thing that we have no idea about the reason why the model predicts the right IC50. So I start questioning about the black box question. For example, when we knock out a gene or treat the cell with a certain drug, how does the perturbation propagate at the gene level in the cell? So existing methods in this domain basically fall into three groups. So the first group, data-driven methods, use the conditional encoder-decoder architecture that purely learning from the data. And here the condition is encoded from the perturbation information. Then knowledge-driven methods like Gears T-experts, in which bring in a knowledge graph as a biological prior for better representing the perturbations. And more recently, semantic-driven methods like GPT that use language model embeddings to represent the perturbation conditions. So currently, most models rely on the MSE loss to reconstruct perturbed cells states, but when we looked under the hood, we identified a major pitfall that we call mean collapse. Here is the example. In a typical perturbation of 500 genes, 5,000 genes, only about 10% are actually differentially expressed. So to minimize overall loss, the model predicts everything close to the control state because it gets a perfect score on the other 90% of the genes. So this is a problem because DEGs are defined by their difference from the control. As you can see in the figure, while the baseline's DEG predictions tend to collapse to the zero, but AdaPert, on the other hand, actually captures the real biological shifts, and we think that's what biologists really care about. And let's take a look at why these methods fail in recognizing the biological signals. As you can see on the left, pure data-driven methods only see the raw gene expression of the control and perturbed cells, so they have no way to tell which genes are important for this perturbation, and they cannot separate the signal from the noise in the massive data. On the right, recent methods try to add a knowledge graph to bring external knowledge into the model, but the graph structure is fixed to every perturbations. So whether you are knocking out TP53 or BRCA1 gene, the model sees the same connections, which may include the external noise. So what we need is a knowledge graph that can dynamically adapt to each perturbation, and that's the core idea behind the AdaPert. So this is AdaPert. The idea is pretty straightforward. We take two inputs. The one is the control cell expressions X of C and perturbation gene P. Then, instead of using the entire knowledge graph, we extract a subgraph that is conditioned by the specific perturbation P. So each perturbation gets its own subgraph G of context. Only the relevant genes are kept. And from that, we predict the perturbed expression X of PERT. So there are two key ideas here. One is the perturbation-conditioned context, which means that every perturbation learns a different subgraph from the knowledge graph. The other is adaptive signal-noise separation. In this part, we use three loss functions that work together to separate real DEG signals from background noise in the training steps. So let me walk you through all of these in the next two slides. So the first, how do we extract this subgraph? First, we get structural embedding from GNN message passing on the knowledge graph. And then we get semantic embeddings from gene descriptions, which are generated by large language models using the NCBI database as a reference. Then we concatenate both structural and semantic embeddings, pass through an NLP layer, and score each gene's relevance to this specific perturbation. Finally, we sample a sparse subgraph in a differentiable way, which are being fed into the decoder and generate the perturbed expression. And on the training side, we use three losses. So the first loss is standard MSE component, keeping all over reconstruction stable. And second is Hilbert penalty on non-responsive genes, which reduces the force changes in the data. And the last one is cosine alignment loss that guides the subgraph to capture the real perturbation effects. So the important point is that different perturbation has different effect size, and by adaptive learning with these losses, AdaPert can learn to treat different perturbations with either strong or weak suppression on reconstruction step, and can generalize to the new perturbation settings. Now let's look at the results. This table shows the overall performance across different methods and datasets. You can see AdaPert improves on almost metrics, and the PDS improvement is larger than Pearson improvement. So the PDS measures how well the model can actually discriminate between different perturbations. So this metric tells us the AdaPert is not just reconstructing better, it's actually distinguishing perturbation effects from each other. And finally, the results I really care about are the DEG level metrics, because in the real world, that's what actually matters to a biologist. On the DEG overlap score, we improved by 20% over the TXPERT. We also outperform every other method on direction match, Spearman correlation of fold changes and the significance score. These numbers proved AdaPert is capturing the real biological changes. We are not just fitting background patterns, we are actually understand the perturbation signals. Well, AdaPert works well. It made us think about a much serious problem in our field. So everyone is talking about big data and foundation models in single-cell biology. However, scaling up hasn't really worked for single-cell domain yet, not like it worked for AI in text or images domain. So actually, I think the reason may lies on the data. Look at the repology dataset with 100 and 1,000 cells, which sounds a lot, but they all come from just 2 cell lines. We are scaling up the same experiments over and over, but we are not scaling the variety. As Bo Wang said at GTC 2026, we need context-dependent scaling. The real bottleneck isn't the amount of the data, it's understanding the context, and that is a problem that no AI researcher can solve alone. And next, I'd like to introduce the collaboration opportunities I am seeking. On the AI side, I can provide our perturbation models, DEG-aware evaluation tools, an LLM agent for biological workflows. And on the experimental side, I am looking for partners with single-cell or bulk cell perturbation data, especially from diverse cell lines or conditions. There are 3 potential directions. First, we can use our model to make predictions, and you can test them in the lab. Second, we can use your new data to help AI work better in different environments. And third, we can work together to define what a good prediction actually looks like for a biologist. In summary, mean collapse is a real hidden problem in our field. AdaPert solves this by focusing on the actual biological signal and separating it from the background noise. But the bigger challenge is making these models work across different cell types, which is something we can't do alone. We need AI researchers and biologists to work together. So thank you so much for your time, and if you are interested, please reach out through the AI.CRED website. Thank you very much.